you'll see RNA with all these little lowercase letters in front, little letters in front. And those are identifying different functions of the RNA to tell one function from another. The first one we saw there, that was messenger RNA. With that, we put a little letter M in front. But then we have, for now, we're looking at two other kinds. There's ribosomal RNA, and that actually becomes part of the structure of the ribosomes. The ribosomes themselves, these workbenches for building proteins, are mostly made out of RNA. So there's an example of a very important activity that RNA does. It doesn't just sit there the way DNA does. So you have these ribosomal RNA molecules that go into building the different ribosomes. So that's little r RNA. Also, we have many different kinds of what are called transfer RNAs, tRNAs. And when we are actually building a protein in the ribosome, it's this coordinated dance between messenger RNA, ribosomal RNA, and tRNA that's getting the job done. All three of those are coming together in the ribosomes. This is an example of a tRNA right here. So what happens with a tRNA, it starts out as a single strand of about 80 or so nucleotides together. So if you just think about having a gene for a tRNA that's located somewhere in a chromosome, it'll have a short region where it's specifying making about an 80 or so nucleotide sequence. And some of those nucleotides get modified. Notice the ones with the red dots. Those are altered. They're not the standard ones anymore. You started out making a transfer RNA copied from DNA, but then it gets altered some. And so we've got some changes going on. Here's a different base called inosine that's put in there. And this one does not, okay. Notice that we have a 5' end and a 3' end, just like we would have with any transcript. We're going to go and copy it, and it'll have these two different ends. And in this case, it's folded back on itself, forming base pairs in 4 regions. So what you'll end up having is all this base pairing between C's and G's. Here's an unusual spot with this one with a U and a G. There are a U and a U that are not actually pairing with each other, but then the C and G there does. Short base pair sequence, short base pair sequence, again with some kind of weird stuff going on. And what we've got is one end of this tRNA carries an amino acid, one of the 20 amino acids. And at the other end, we have an anticodon that is able to actually pair up with information that was coming from a gene. The tRNA has 2 ends. One of them is an anticodon end that can pair up with information from the DNA. The other end is carrying an amino acid. So what you get is information from the DNA pairing up with an anticodon and the correct amino acid being brought in by this entire complex. This is a very complicated looking thing with some odd stuff happening in it. There are many different versions of these tRNAs that have been produced with very subtle differences in properties to basically do correct pairing here with information from the DNA and correct binding to the right amino acid at the other end. So what we have with these RNA molecules is that they're behaving in very sophisticated ways to help build the proteins. Well, again, in this case, with nucleotides being modified to make subtle changes in their behaviors. Here's part of a ribosome. This is the large subunit. Ribosomes have a large and a small subunit that have to come together for them to actually start building a protein. This is the large subunit. Notice the yellow and the gray stuff that's in here. All that gray stuff is actually ribosomal RNA. So this has proteins that are shown there in yellow. So proteins are playing an important role in the ribosome's activity. but the great bulk of it is actually RNA. So this whole structure that is building proteins is mostly made out of RNA. To give you a sense of how dynamic a molecule RNA is. So when you hear the term enzyme, you should normally think that it's a protein, but you also have enzymatic RNA molecules. We call those ribozymes to tell them apart from the normal enzymes. Enzymes are almost always proteins, but with this whole thing with protein synthesis, we get these exceptions, and those would be the ribozymes. So the ribosome is where we're building proteins, and the ribosome is about 80% RNA. The rest of it's protein. Here's where the ribosomes are actually being made. You have genes for them on some of your chromosomes, and so if you're looking inside of a cell, maybe the cell is this big, you have the nucleus, and then you have that dark region often within it. That's the nucleolus. That's where the cell is busy making millions of new ribosomes. Something like a liver cell probably needs about 5 million ribosomes. If a liver cell is produced from an earlier cell and then it splits and produces 2 new liver cells, well, right now they're probably starting out each with 2.5 million, and they need to make 2.5 million more, so they get busy. They have this nucleolus, which is then producing new ribosomes, and what you get are these DNA strands within the nucleolus, DNA molecules that have the genes on them for making the ribosomal RNAs. And here we have many copies being made of the ribosomal RNA. Notice these feathery arrangements. All these molecules coming off the sides are copies of the ribosomal RNA. Here's another one. And here we've got very short RNA molecules because they're not done yet. But down to here, we've actually made much longer ones. Here, we're nearly done building the new copies of the ribosomal RNA. That's as we are coming along here with RNA polymerase, and it's building many, many copies of the gene for making these ribosomal RNA molecules. So one gene that's on a chromosome here is being read simultaneously by many messenger RNAs that are grinding along it here, and each one is producing a copy of that gene to eventually get the ribosomal RNA molecules that go into making the ribosomes. This is all happening inside the nucleus. That's how you're able to go and quickly make millions of new ribosomes, because here in this picture we're making thousands of new ones at once. Then similar activity goes into making these tRNAs. You have genes for encoding the tRNAs, which, as we saw, are around 80 or so nucleotides long. And each tRNA will form that cloverleaf arrangement that we saw earlier, but it actually folds up into a more complex, twisted shape with then these different base paired regions within there. Within different parts of the tRNA, it's trying to form a double helix anytime it's base paired, like we see here, here, here. And at one end of a tRNA, it will bind to one and only one type of amino acid. There's many different kinds of tRNAs. Whichever one you have, it's only going to bind to one particular amino acid. It'll reject all the others. And at the far end of it, down here, we have the anticodon end. This will form a base pair arrangement with a codon that's on a messenger RNA. So if you have a messenger RNA and it's got 3 letters on it that were encoded from a gene, there will be an anticodon that pairs up with that. And if it pairs between the anticodon and the codon, that means that the tRNA brought in the right amino acid to go with that codon. So these are the actual links between the 2 languages we talked about. We are going straight from an amino acid language here, written in 4 letters, to an amino acid language here with 20 different possibilities. The tRNAs are the bridge between the two, and they do this right inside the ribosome. So what you'll have is a 3-letter sequence at one end, which is able to form a base pair arrangement with a messenger RNA. And then at the other end, you have a particular amino acid connected, which becomes part of a polypeptide as the ribosome assembles it. So different things happening at opposite ends of the tRNA. And in our cells, we have many different kinds of these because, at the very least, we have 20 different amino acids, and you need a different tRNA for each type of amino acid. Now, inside the ribosomes, you have this arrangement where once the ribosome gets busy actually translating and building the protein, it has the small and large units put together, and there are 3 binding sites inside it for tRNAs. It also clamps onto the messenger RNA. Messenger RNA has the information for making the protein. There it is. It spools through the ribosome, and there are 3 binding sites for tRNAs inside the ribosome. And these tRNAs keep bringing in amino acids and forming base pair arrangements with information that's on the messenger RNA. So if you have 3 letters right here that are specifying, say, a lysine amino acid, there will be a lysine that's brought in that pairs up with that. So inside the ribosome, we have these ribosomal subunits, large and small. They attach to the messenger RNA near the beginning of it, and they have these 3 binding sites. tRNAs have these 3-letter sequences that we call anticodons. They will pair up with codons that are on the messenger RNA. You get pairing between codons and anticodons. So for instance, U's there would pair with A's there, C's there would pair with G's there if they line up with each other. So you can get a weak connection, hydrogen bond connection, between the anticodon that's on the tRNA and the codons that are on the messenger RNA. For example, U's pairing with A's. And if this is the situation, we can go back to that genetic code table, and we can see that this codon on the messenger RNA that reads AAA, that calls for lysine, one of the amino acids. If you have this codon, then you have to have a tRNA that has this anticodon on it, and it will have a lysine on the other end attached to it, ready to bring the lysine in that this is calling for. So the ribosome builds up this whole sequence of amino acids based on these 3-letter groupings that are coming in the messenger RNA. Each 3-letter grouping is specifying a particular tRNA that has a particular amino acid attached to it. So this is what the messenger RNA is bringing in, these 3-letter groupings, and you're getting different tRNAs that come in that are able to base pair with these and bring in, for instance, proline. If a tRNA comes in that has an anticodon that matches with this, that is complementary to it, so the anticodon could be GGC that pairs up with this. That tRNA will also have that amino acid attached to it, and no other kind. So there's pairing between codons and anticodons. The anticodon is complementary. They pair up inside the ribosome, and the tRNA is going to bring in reliably the correct amino acid that pairs up with the 3 letters that were on the messenger RNA. So initially you have one subunit of the ribosome floating free in the cytoplasm, that subunits only come together when they're actually busy building a protein. Otherwise, they separate from each other. Here we have the small subunit. It's floating free in the cytoplasm, and it has this initiator tRNA attached to it, along with the first amino acid, the methionine that goes with the start. It's energized by this triphosphate GTP, basically the same energy as ATP, slightly different molecule, using a G base instead of the A base, but otherwise the same thing as ATP. So you have this sitting in the cytoplasm. If there is a messenger RNA that is able to receive this small subunit will attach at the 5' end where the cap is, and it will scan along until it encounters the very first AUG. That's the codon that calls for methionine. That's also the start codon. And so that tells this, here's where you're going to start making a new protein. So the small subunit slides to there, stops. Then we have to build the rest of the ribosome around this before it can proceed. So the large subunit attaches. We end up using the energy of the GTP, so now it's released. And we have this initiator tRNA along with this first amino acid sitting in the P site, the peptidyl site. But here we've got the what's called the amino acyl site, and over here exit. This is all ready to go now. So now we can start bringing in more tRNAs, bringing in more amino acids. So once we got that set, we had the initiator tRNA in the very first position on the first AUG. And now we can bring in another tRNA, and it will have to have an anticodon that pairs with this codon. Maybe this is CCU, and so this is going to be what? GGA in that case. You'll have base pairing between the anticodon and the codon. A particular tRNA comes in that matches this codon, and it's guaranteed to be bringing in the right amino acid that goes with that codon. That pops into the amino acyl site, the A site, and you end up then grafting whatever you already had onto the new one. There is a transfer, taking the amino acids that were here and popping up onto the new one that came in. So now the chain gets longer. Then the ribosome moves on 3 bases. It slides 3 bases down, and these slide from the A and the P sites over to now the P and the E sites. This tRNA has now been used. It's ready to be ejected. So it is then spit out. It can bind to another amino acid and repeat the process, be available to do this again. And once again, we have an open A site. So now we can bring in another tRNA with another amino acid on it. That starts right here. That's the first AUG when we're looking at the whole mature transcript. It runs along just putting in one amino acid at a time for every 3 bases, for every codon, until it gets to one of the termination sequences. There's 3 of those. This is one of them. So you're busy. We've already now built this polypeptide going on for a ways. It began with methionine, and now we've got a lot more amino acids in there. We're still ejecting empty tRNAs, bringing in new ones, attaching this polypeptide chain onto whatever's new coming in, and then the ribosome moves on 3 bases to repeat the process. And it eventually gets down to one of the termination sequences. Imagine this is one of the 3 terminators. Now we get what's called a release factor that binds to that. There's no tRNAs that will bind to one of the terminators. Instead, a release factor does. And once the ribosome gets to that, it triggers the release of the polypeptide, and the ribosome falls apart into two parts, releasing all the rest of the hardware there. So now we've created a polypeptide, and it's free in the cytoplasm to do whatever happens next to it. Now, you can have many ribosomes working on one transcript at a time. Here we have a long RNA transcript, messenger RNA, and here's numerous ribosomes working along it, all starting at the 5' end and then proceeding down it. So we're making many copies of the protein simultaneously. That's what all these are. These are the polypeptides. They're growing. They start at one end and work further and further along, each of them chasing after the ones ahead. So you can get something like this, a polyribosome. This is the 5' end. That's where the cap is. That's where you start the process. The ribosome attaches, begins building the polypeptide, and keeps going and going. And notice the chain is getting longer and longer. And eventually it gets to the stop, disassembles, releases the polypeptide, and there's many more chasing after that one. So you can make many copies of the protein at once, depending on what the needs of the cells are, because we'll see a lot about gene regulation later in a later chapter. But the potential is there to make many copies if that's what the cell needs. So altogether, you can think about how you have all these chromosomes, and they contain genes for making, of course, the messenger RNAs. That's what you normally think of as genes, making the messenger RNAs, but also genes for making ribosomal RNAs, and for making all these different tRNAs, lots of them. And so you end up producing all these different tRNAs, these different subunits of the ribosomes. You put those together to then build up the ribosomes along with the proteins that go into in with the ribosomal RNAs. You get the different kinds of tRNAs in the cytoplasm. You get the messenger RNAs that are going through alternative splicing to remove the introns, put the exons together in different arrangements. And here we end up then having messenger RNA going right through a ribosome and building protein. Let's watch a short film that shows translation, and then we can get on with an activity. A short film first. It's another one of these high-quality ones. Translation right here.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape. The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a three-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides, or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
I'll put that and the other video I showed you into the Chapter 15 lecture folder, so you'll be able to see these films in Canvas. Now, I'm going to show just a couple of tidbits here, and then we'll get on with this. Looking at the tRNA, I mentioned how some of these bases are modified. That gives the tRNA some very specialized behavior. Down here, you have a base that's actually modified on this one into what we call inosine. That's one of the modifications, which makes it so that anticodons don't have to exactly match the codons. You might think that there's 64 different codons, right, as we were saying here. How many of those are actually used to pair up with amino acids? Is it all 64? No, because you have 3 stop codons. That leaves you with 61. You might think then that you would have, if you got 61 different coding codons, that you would need 61 different tRNAs to pair with those. But what usually happens, or what often happens, is that when you're looking at the anticodon of a tRNA, the first 2 letters usually make an exact match with a codon, but the third one is sometimes variable and can kind of bridge the gap between 2 different third letters on a codon. This inosine, as they're saying here, it's able to form 3 different base pair arrangements. That means that this could actually pair up with 3 different codons. And so you have these tRNAs with some modified letters, some modified nucleotides that are a little more flexible. And it's called the wobble hypothesis. That was something Francis Crick from Original Discovery of DNA came up with. The idea that even though you'd expect to have 61 different tRNAs, there actually aren't that many. With us, it's more like 40. In humans, there's about 40 different tRNAs that we use. And it's because you don't have to get an exact match between every anticodon and every codon. Some of them, you actually have these flexible anticodons that are able to pair up. As long as they get 2 of the bases right, they're able to kind of pick up the third one too. So I have a question for you on the group discussion that's related to that. Anyway, it's not quite as many different tRNAs as you would imagine because of this wobble. You'll see some about that in your chapter. And there was one other thing here I wanted to bring up. If I don't find it, then we'll just be on our way here. Yeah, I'm not finding it. So let's get on with the group discussion here. And if I think of this third thing, I'll bring it back up.